Hello everybody, my name is Verena. I'm a PhD student at the University of Kaiserslautern. I will tell you about predictions in marine biomonitoring using supervised machine learning. I try to find out how much data is actually needed to still make good predictions on environmental status. First, I want to give you a short reminder on marine coastal ecosystems. They offer numerous ecosystem services and are at the same time exposed to a variety of stressors from human activities. They are exposed to pollutants from industry, tourism, and eutrophication. Further, they can be influenced by commercial shipping. Commercial shipping can introduce invasive species and also pollutants. Also, it is known that aquaculture can have a huge impact on the bacterial community on marine coastal sediments. If you're interested in the underlying ecology, please listen to Larissa's talk tomorrow in the session of Working Group 2. Because of the huge impact we have on marine coastal ecosystems, environmental biomonitoring programs for effective management and conservation are crucial. Studies characterizing those environments using eDNA metabarcoding have become more and more popular. The transition from traditional macrofauna-based monitoring towards eDNA metabarcoding-based monitoring is fortunately in progress, as it can save lots of money and time. The basic idea is that the eDNA of the microbial community can mirror the occurrence of traditional bioindicators. In recent studies, this eDNA was sequenced to full extent, which results in very large datasets. In our study, we focused on those datasets for environmental impact assessments. The aim of our study was to find out how much data minimum is actually needed to perform valuable predictions while optimizing sequencing times, sequencing costs, and computational requirements. To reach this goal, 4 different datasets were analyzed in total. 3 datasets were already published in the literature and were reanalyzed. The first dataset we used was published by Gerhard and Gunsch, who performed a machine learning study on ballast water in different harbors of the world. We used this dataset to predict the country of origin of the water samples. The second dataset was published by ATSTI in 2017. In this study, they developed a microbial marine biotic index, which is an extension of the traditional index. We used this dataset to predict environmental quality. The third dataset was published by Tristan Cotier, also in 2017. In the study, the ecological quality status of marine environments is predicted using supervised machine learning. Because this dataset is huge and the samples were taken at different salmon farms, not only the ecological quality status, but also the farms of origin were predicted in our study. The fourth dataset is novel and was processed in our lab. It's based on V3, V4 amplicons from Scottish salmon farm sediment. As prediction objectives, the distance from the fish cage, the station, was chosen. Further, we are happy to have samplings from different time points, so we could also predict sampling season. Now I want to give you a short overview of the basic idea which was applied for all datasets. Here you can see a classical rarefaction curve. I am sure you have all seen one before. With all available sequences, supervised machine learning models were constructed. We used the random forest classification approach with different combinations of parameters. ASVs were used as features. The mentioned prediction objectives were used as labels. Let's zoom in on this a little bit. Now, a traditional rarefaction was conducted using minimal sequence number. So in this example, all samples are reduced to approximately 15,000 sequences. With this subset, machine learning models were constructed again. Now a new thing happens. We further downsampled the sequences. This means that for each sample, sequences are reduced to, for example, 12,500 sequences. With this new dataset, the random forest models were constructed again. After this step, all the samples were then reduced to 10,000 sequences. And again, models were constructed. And so on, and so forth. So, with every set of supervised machine learning models, the number of sequences per model decreased. To find out how much data is needed for making accurate predictions to random forest model performance was compared between the original full model using all available sequences and the models based on less sequences. Exemplary, here you can see the results for the novel dataset. The blue line indicates the decreasing model performance. Also, the colors show the range of the kappa value reached for each model. Kappa more than 0.8 indicates almost perfect agreement. For the prediction of the station, the random forest prediction accuracy decreased steadily with decreasing number of sequences. In this example, kappa value drops below 0.8 if less than 5,000 sequences per sample are used to make the prediction. For the sampling season, accurate predictions could be made even when the number of sequences decreases heavily. In this example, the far left models are built on only 50 sequences per sample. This means that only 50 sequences can accurately predict the sampling season, which is amazing. Those analyses were repeated for each dataset. The minimum number of sequences needed to obtain good predictions ranged between 50 and 5,000 sequences. We can frame the first take-home message. Even in the worst case, 5,000 sequences minimum are needed for the prediction. Because both 50 and 5,000 sequences is the threshold within the same dataset, the sample number cannot be the discriminating factor to know how many sequences are needed for the prediction. For both prediction objectives of our novel dataset, we have 3 categories each, so also the number of categories cannot give a hint on how many sequences we need. Another thing which comes to mind is the sample distribution among categories. If you have an uneven sample distribution, chances are naturally high that a new sample will be classified in this category. To exclude this factor, the distribution coefficient Gini was calculated for each category. The Gini index is not to be confused with Gini impurity. The higher the Gini coefficient, the higher is the sampling imbalance. For both groups of samples in our dataset, the Gini index is quite similar and also quite low. Therefore, in our case, the sample distribution cannot give a hint on how many sequences we need. But what are the factors for separation of categories? A first indication for the number of needed sequences could be feature specificity. If you take station as a category, you can see that nearly 35% of all features are shared. Station B only shows about 6% of unique features it does not share with the other stations. If the features are grouped by season, only around 11% of all features occur in every category. Spring and summer each show 10% specific features. We can hypothesize that the more features are shared between groups of samples, the more features are needed for making good random forest predictions. Another idea was to look not only at the occurrence of features, but also at the variation of abundance. Therefore, the coefficient of variation was calculated for each feature, first categorized by station and second categorized by season. The CV measures the standard deviation of the feature abundance across the group mean. The average CV of these features categorized in stations is 1.2, whereas the CV based on seasonal grouping is higher. This could also be a hint on further forecasts on how many sequences are needed for making accurate predictions. Because we try to distinguish between categories, the separation of the samples within these groups could be relevant. An easy way of representing similarities between samples is a multidimensional scaling. In this case, the seasons are colors and the stations are shapes. On first sight, you can see that the colors are more separated than the shapes. For statistical significance, you can look at 95% confidence intervals. Based on station, you can see a huge overlap, which means that there is no clear separation between the categories. On the other hand, if you group the samples by season, you can see a nice separation. This separation ability of categories might give an indication of how much data we need. And this is our second take-home message. Let's come to the conclusions. For our tested datasets, even in the worst case, 5,000 sequences were enough to still make predictions as good as using the whole datasets. The minimum number of sequences we need to make equal predictions seem to rely on the ability to distinguish between the categories. Characteristics as number of samples, number of categories, and sample distribution of categories seem to have no impact on the ability to distinguish between categories in our dataset. On the other hand, feature specificity and feature variation in abundance among the categories could play a role in determining how big the sequence dataset has to be. Additionally, the separation of categories based on sample similarity could play a role in foretelling how much sequences are needed for making good predictions. Further to-dos are now testing of gained information on other datasets to confirm or disprove our gained results. And of course, you can improve supervised machine learning methods per se by, for example, eliminating highly correlating features. And of course, ask smart colleagues for more ideas. And if you are further interested in the recently submitted results, I am happy to keep you updated once published. And with that, I want to thank the scientists which published the investigated datasets. I am happy for financial support by DFG and Scottish Sea Farms. And I want to thank my colleagues Timo Mühlhaus from Computational Systems Biology and Tom Wilding from the Scottish Association for Marine Science. Further, I want to thank Thorsten and Larissa from my group for supervision and data acquisition.